right, guys, let's bring it moving on here. Let's bring on Eric Richman. He's the CEO of GANX Therapeutics. There he is, ticker GANX. And Eric, we've got your slides. I see them. We'll bring them up on the screen. There they are. I'm going to step backstage here and give you the floor.
Spencer, thank you very much, and good morning, everybody. It's really a pleasure to be here at your conference, and appreciate the opportunity to introduce you to GANX Therapeutics. This company, GANX, is leading the discovery of finding novel binding sites to create new medicines for diseases with unmet medical needs. Our therapeutic focus is in the area of lysosomal storage disorders and central nervous system disorders, including various neurodegenerative diseases. We're developing small molecule treatments, and the way that we do this is all based on our platform technology. Our platform technology is patented. It's a physics-based approach. It's based on molecular dynamics, and it can very rapidly, very efficiently, and very systematically identify previously unknown binding sites on proteins that have been associated and very well validated as the cause of various diseases. And I'll get into that in just a moment. But these binding spots on these proteins are called hotspots or allosteric binding sites. An allosteric binding site means that it can be a regulatory site, which means it can either turn on the protein function or it can turn off the protein function. Now, this platform technology, which has been about a decade in the making and patented, has given rise to our internal product candidates, as well as a pipeline of products that we have partnered with various pharmaceutical companies. So we announced a partnership last year with a large Japanese pharmaceutical company called Sumitomo Dainippon in the area of demyelinating disease. And also in the area of oncology, we announced a multi-target deal with leading biotech company Zentalis Pharmaceuticals. And I'll get into that in just a moment. But first, a little bit of understanding about what is a non-druggable target. Now, there are many proteins that are associated, very well validated, well understood, that certain proteins that when they are defective or in the wrong conformation or what's called misfolded, they cause various diseases. And these diseases range from cystic fibrosis to oncology to metabolic diseases to neurodegenerative diseases. And it's very well understood. Protein is defective, causes a certain disease. But the difficulty has been finding a spot on that protein to bind to, which makes that protein druggable. And only about 10% of those proteins that we know cause disease can actually be targeted. So what our technology does, and with the recent advances in computational biology and supercomputing, our technology opens up this field very wide, and we can now identify spots on these proteins that, when targeted, we can finally make progress and address some of these diseases that have no known treatments. And we do that by targeting these hotspots or allosteric binding sites, which allows for restoring protein function. Now, this is very specifically the problem that we're trying to solve. In the center of this cell, you see a misfolded protein. This is the root cause of many different diseases, as I just mentioned. It's misfolded because of a genetic mutation or a stress factor. And that misfolded protein, because it's in the wrong conformation, cannot function normally. And it gets stuck in the cell, can't move through the cell, it can't work on the substrate, and therefore the substrate builds up in the lysosome. That's what causes the symptoms and the disease. Our technology you see on the right side, where we create small molecules, we can identify the allosteric binding site or never previously described site on this protein, create a small molecule that binds to it, and we refer to those as stars, or structurally targeted allosteric regulators. And these stars or small molecules bind to that protein, stabilize the protein by putting it into the correct conformation. And once that happens, the protein can then move through the cell, it can work on substrate, and reduce toxic substrate accumulation. Now, our focus, again, is on the allosteric binding site, and that's separate from the active binding site. This is a picture of a cartoon of an enzyme. And you'll recognize this as the active binding site where most drugs, almost all drugs, bind to that active binding site. But we're not competing with drugs. We're actually targeting the allosteric binding site, which leaves this space open and available for what's called wild type activity. And that offers a broader therapeutic window. So at Gain Therapeutics, we're generating value in two areas. Number one, our business development engine, which is our discovery platform. And what are we doing with this platform? This produces novel targets. This is what drives the biotech and pharmaceutical industry. Every company is looking for new targets, new ways to treat diseases. Some diseases have never had treatments before. That's why our technology platform is so important and so valuable. Secondly, we have a diversified pipeline of products that we have developed internally that all came from our pipeline. From our platform. So if everybody understands that certain proteins are associated with certain diseases and all you have to do is bind that allosteric binding site, why doesn't everybody do this? The reason is because it's extremely difficult to find that allosteric binding site. And we have a platform technology that does it very rapidly, very efficiently. And we start with the 3D structure of the protein that we know is associated with the disease. And with our algorithm and powered by supercomputers in Barcelona and Lugano, Switzerland, we can process to several 100,000 calculations per second to model how the surface of this protein can produce free energy, changes in free energy. So we're looking for delta G changes or changes in free energy, which is a physics-based approach to being able to find out which of these sites on the surface of the enzyme and within the folds of the enzyme and within the crypts have the potential to be regulatory sites. And once we do that, we screen up to 10 million compounds with the supercomputer and to find which ones have the closest fit. And then those come down to the bottom of the funnel, and then we move those through our medicinal chemistry process. Now, this is not trivial. This is something that can shave years off of the drug discovery process. This is done in 4 to 6 weeks. And typically it can take years to identify an allosteric binding site on an enzyme. And we have identified allosteric binding site on proteins and enzymes that have never been described previously. And this is really what is the power of this technology and why it's so transformational. So we've done this several times. It takes 4 to 6 weeks to identify allosteric binding sites, about 2 months to screen compounds, and then we move into moving these scaffolds or compounds into drugs. Now, our company, we began in the area of lysosomal storage disorders for a few reasons. Number one, unmet medical need. There's about 80 or so lysosomal storage disorders described in the literature. Only 5 or 6 have any treatments whatsoever. Those treatments, none of them cross the blood-brain barrier. They're all large molecules, enzyme replacement therapy. They don't cross the blood-brain barrier, and therefore they cannot treat the neuronal symptoms of these diseases. And these neuronal complications are very severe, and they're seen in almost all lysosomal storage disorders. So that's the reason number one, unmet medical need. Number two is the market is very, very, it's a very strong market. It's a very big market. It's about $6 billion, despite the limitations in drugs available today, and it's growing rapidly. Number three, our strategy is to identify lysosomal storage disorders that have a direct link to more prevalent indications. So, for example, the most prevalent mutation that is found in Ashkenazi Jews, for example, that's one population, is a disease called Gaucher's disease. And that mutation is a mutation of a certain gene that causes a defective or misfolded enzyme. And that misfolded enzyme ends up causing Gaucher's disease. That exact same mutation is also present in up to 10% of Parkinson's patients. So by us developing a treatment for Gaucher's disease, and I'll tell you about this program in just a moment, we have a direct path to having, to be able to impact more prevalent diseases like Parkinson's disease. And in the future, the platform is agnostic. We can develop, we can find out allosteric binding sites in many different diseases and therapeutic areas. And that's demonstrated by our relationship with Zentalis Pharmaceuticals, a multi-target deal in oncology. So when we go through that process, we scale it down to find a couple of drug candidates to move forward. We have a few criteria that's very important. First of all, it has to target the majority of mutations, and that means it has to be in excess of 65%. It has to cross the blood-brain barrier. All of our strategies, our treatment strategies, are oral, so it has to be able to cross the blood-brain barrier after being taken orally, and also have to stabilize the misfolded protein and reduce toxic substrate. And I'm happy to report that with all the in vivo and in vitro data that we have generated so far, all of our programs accomplish this. So now I'm going to highlight, this is our pipeline. I'm going to highlight our lead program, which is the Gaucher Parkinson's program. As I mentioned, these two diseases are linked because they share the same genetic mutation. Up to 10% of Parkinson's patients have the same genetic mutation as what causes all three types of Gaucher's disease, type 1, type 2, and type 3. Gaucher only has a treatment for type 1, but again, it doesn't cross the blood-brain barrier, so the neuronal complications are not addressed. Type 2 and type 3, which are neuronopathic Gaucher's disease, have no treatments whatsoever. Now, the mutated gene causes a defective enzyme called GCase, and that enzyme results in a buildup of substrate. That's what causes these devastating symptoms in Gaucher's disease. In Parkinson's disease, the same genetic mutation causes a defective enzyme, a misfolded enzyme, that enzyme that can't work properly. And because it can't work properly, it causes a buildup of the hallmark of Parkinson's disease, which is called alpha-synuclein. And I'll show you some of the data here. In the interest of time, I'm not going to go through this, but we have all of this data on our website. Basically, our compounds can be found in the plasma in a dose-dependent manner, and also in the striatum, which is a section of the brain where you observe Parkinson's disease. This data was presented at the World Conference on Parkinson's disease in May, which is funded by the Michael J. Fox Foundation and also the Silverstein Foundation. And what this data showed is something very important. It showed that we had positive effects in all three of the industry-accepted assays for characterizing whether or not a product candidate should move forward in Parkinson's disease. And I'm very briefly going to walk you through this data. This is in rats, and these are normal rats in the black, and they have very low levels of alpha-synuclein, which is the hallmark of Parkinson's disease. When they are given a chemical called rotenone, they have a Parkinson-like syndrome that is induced, and you can see exactly what you expect, an increase in the hallmark of Parkinson's disease, alpha-synuclein. When these rats are given our compound, there's a return towards normal in the level of alpha-synuclein. So we can actually reverse the neurodegenerative effects of Parkinson's disease in this model. Secondly, we looked at neuronal activity, and you see the same thing. You see an improvement in neuronal activity after these rats have been given Parkinson's disease with this chemical rotenone. And then the third question we asked is whether or not the animals have any improvement in locomotion, tremors, the ability to walk distances, and the answer is yes. We saw an improvement in locomotion in these rats. Now, this was very exciting data. This was awarded top 10 presentation in May at the World Conference on Parkinson's disease, and it was very encouraging. However, it's an animal model, and there's limitations in these animal models. So the next step was to look at our compounds in patients, cells that are derived from patients who suffer from Gaucher's disease and Parkinson's disease. And this model was developed by a key opinion leader, Dr. Ricardo Feldman at the University of Maryland. And this is thought to be the closest approximate of what happens in human clinical studies. So this is a very important data. It's the step between animal studies and human clinical studies. And we were very pleased to report in this model, and we announced this about 2 weeks ago, that just top-line data, that our compounds had very marked results and very marked improvements in the key biological readouts. So it showed that we were, first of all, our compounds were able to increase the enzyme activity. We were able to increase the amount of enzyme. We were able to reduce the toxic substrate associated with Gaucher's disease. and also deplete alpha-synuclein, the hallmark of Parkinson's disease. This is exactly what we expected to see, and we're very pleased to see it. But also, most importantly, we were able to show very clearly that our compounds helped the defective enzyme co-locate into the lysosome. This is very important because this is what distinguishes our compounds from other companies and other candidates that are out there that are trying to develop the same sort of data. So having the enzyme, observing that the enzyme is transported to the lysosome shows that the enzyme actually can avoid the protein degradation, a quality control mechanism within the cell. Very, very important. It's a key differentiator for our technology and our compounds. So in dopaminergic neurons, our compounds decreased alpha-synuclein levels. This data was presented, just the top line. It was, we were invited to present this data at the Michael J. Fox conference on November 10th. So please stay tuned. Please listen and look for that data presentation because what you'll see are all the details supporting these claims about very positive data. So we're very excited about moving forward with this program. Of course, all of our compounds are patented. We were issued patents earlier this year for the GBA program, not only for Gaucher's disease and for Parkinson's disease, but also that enzyme has a role in Lewy body dementia and also Alzheimer's. And so those patents were filed for those indications. So upcoming milestones, we will be initiating IND-enabling studies this year and filing IND next year. We hope to be in the clinic next year with our Parkinson's program, at least in first time in human studies. This is a very exciting program that we're looking forward to getting into the clinic. Our other programs targeting lysosomal storage disorders are on a very similar timeline, IND filings next year. What's not in here are our partnerships and the progress that we're making with our partnerships, especially Zentalis, where we have identified targets in oncology. You'll see in our Q that we're generating revenue from those, from that collaboration on a quarterly basis. And we are entitled to milestones related to regulatory events, clinical milestones, commercial milestones, and also royalties on commercial sales for up to 5 targets in oncology, which can be very valuable to us and our shareholders. We completed an IPO in March of this year. We raised $46 million. This was 5 times oversubscribed, but we only needed $40 million to get us through the next 2 years. So we thought it would be better to close on $46 million and raise additional capital once our INDs are filed and we have established partnerships and an increase in valuation. So we are very excited about the progress that we've made since the IPO, which is doing 2 corporate deals, which are validation of the technology, and also generating this very important data in the iPSC models. My background is in venture capital and also in early stage biotech companies. Our founder has an unbelievable background in early stage biotech and pharmaceutical companies. All of our directors are independent directors. Great group to work with and very connected in the pharma and biotech world. Our management team equally very well qualified and very experienced. And you can see we've just brought in Terencio, who was the head of CMC at Jazz Pharmaceuticals. As we transition to a clinical stage company, we are bringing in head of CMC, regulatory, head of clinical affairs, design clinical studies, and other important positions to position us well for being able to reach those goals and begin the clinic. So in summary, GAIN Therapeutics is a small molecule company with a discovery platform that is extremely robust, that is potentially transformational because it cuts down discovery time. We can identify novel targets that have been thought to be undruggable and now suddenly are druggable. And we're making great progress, not only with our internal pipeline, where we've been generating great in vivo and in vitro data, but also with our partners, with Sumitomo and also with Zentalis Pharmaceuticals. And also, we have many milestones coming up this year, next year, big value inflection points. And finally, we have a very seasoned management team and board of directors to make it all happen. So thank you very much, and open it up for questions.
Thank you, Eric. This was a really interesting presentation, not just the pipeline, but the discovery platform and the supercomputer. I feel like I need to rewatch that because I tried to understand that, but it was really interesting. And there's a lot to lot to digest there. But would love to learn more about that. We are out of time though for today. So there's the URL up on the screen. Gain Therapeutics. Eric Richman is the CEO. Eric, very interesting stuff. Thanks for joining us today at the Benzinga Healthcare Small Cap Conference.
Thank you very much for the opportunity. Appreciate it.
All right. This next company